Chris will continue our speakers throughout the day, and these leaders will also be joined during the Q&A sessions by additional oncology leaders and experts. So with that, let's get started.
Tonight, I'm gonna have myself a real good time. I feel alive. Don't stop me now. Don't stop me. 'Cause I'm having a good time. Having a good time. A superstar leaping through the sky like a tiger, defying the law. Oh, explode. I'm burning through the sky. Yeah. 200 degrees, I'm smiling, calling Mr. Fahrenheit. I'm traveling at the speed of light. I wanna make a supersonic woman of you.
Welcome, everyone. Welcome. It's a great pleasure to have you all with us here at the Pfizer headquarters. Just over 2 months ago, we actually closed the acquisition of Seagen. And on behalf of the whole leadership team, we're excited now to unveil our new oncology organization to you today. Over the next few hours, we'll do a deep dive into our expanded portfolio, our clinical development plans, and our capabilities. We will also share more about our strategy, which is assigned and designed to drive significant innovation and growth through 2030 and beyond. You've heard a lot now about Pfizer's oncology's renewed focus. As a new organization, we have a bold vision that aligns with Pfizer's purpose. Pfizer's purpose, breakthroughs that change patients' lives. Our vision is to accelerate breakthroughs that will help people with cancer globally live better and longer lives. This drives everything we are doing and reflects our personal mission to improve the lives of people with cancer. Our vision is ambitious, but rightfully so, because as you all know, cancer remains one of the greatest health challenges of our lifetime. For the first time, 2 million people in the US are expected to be diagnosed with cancer this year. Globally, around 20 million people receive a cancer diagnosis in 2022 alone. Approximately 10 million people around the world died from cancer in 2022, and just over 600,000 deaths are expected to occur in the US this year. We believe advancing the global fight against cancer will have a tremendous impact on society. And that's why so many companies in this industry are working towards delivering new breakthroughs for people with cancer. In fact, the oncology market is projected to increase to up to $500 billion by 2030. This year, Pfizer is celebrating our 175th anniversary. We have demonstrated the ability to innovate with urgency, and billions of patients globally have been treated with one of our medicines or vaccines. Seagen has pioneered the technology and development of antibody-drug conjugates, or ADCs, which are emerging as a very, very important tool in the fight against cancer. Over their 25-year history, Seagen developed deep technological expertise to become an industry leader in the ADC field. And today, nearly half of all the FDA-approved and marketed ADCs use a unique linker payload system developed by Seagen. That's why bringing these two companies together has been so exciting for us. By combining our respective strengths, experience, and heritage, we are uniquely positioned to deliver additional breakthrough cancer medicines to more people around the world. As a newly combined organization, our expertise and collective capabilities are now amplified to deliver even more impact for patients than each company could do by itself. Beginning with expertise, we are bringing together the best from both organizations. We have an exceptionally talented team with extensive experience in oncology. Innovation, our pipeline has now expanded significantly, and we are focused on flawless execution, delivering the next generation of breakthroughs. And lastly, scale. We're poised to have a true global impact. Our new oncology organization leverages the breadth and depth of Pfizer, including our industry-leading commercial and manufacturing capabilities, to drive broader patient reach. Before we dive deeper into our future, I would like to briefly touch on what we have been able to achieve over the past decade. Pfizer has been growing our presence in oncology for about 20 years. With the establishment of the first oncology business unit in 2014, we've emerged from a niche player to a bona fide leader today. Leading up to 2023, we established a solid foundation with numerous innovative medicines that became new standards of care in several cancers. In fact, our medicines reached just over 2 million patients with cancer last year, which includes our innovative medicines, biosimilars, and cancer therapies from Pfizer's hospital unit. We've also seen outstanding revenue growth of our oncology business over the last decade at a CAGR of over 19%, outpacing the industry average of approximately 10%. With the acquisition of Seagen, we believe our industry-leading portfolio is positioned for strong near-term growth. We now have 4 additional approved medicines, all of which are first-in-class in their respective indication, and all 4 have demonstrated overall survival benefit. Mid to long term, our early-stage and late-stage development programs have doubled, representing significant future opportunities. We are at an inflection point of our leadership. We expect to impact many more lives of patients with cancer over the next 5 years plus, aiming to double the number of patients treated with our innovative cancer medicines by 2030. Leading up to the close of Seagen, we had several months to undertake integration planning and ensure a seamless transition on day one. The Pfizer Oncology Organization, which includes the US Oncology Commercial Division and end-to-end global R&D, is designed to improve interaction and collaboration across all functions, from early discovery all the way through to commercialization, facilitating quicker decisions and to accelerate execution. Here's our new oncology leadership team. Any organization is only as good as its people. And I'm pleased to say that we're one of the most experienced and talented groups of oncology leaders in the industry, with nearly 300 years of oncology experience across academia and industry between these leaders combined. These are leaders who've done it before, and they have highly experienced teams working with each of them. Retention of talent has been critical for us with this integration, and today we have approximately 50-50 participation of Seagen and Pfizer colleagues in the combined new organization. These colleagues are united around our vision to accelerate breakthroughs for patients with cancer. Our integrated oncology organization is now fully operational. To further illustrate the executional excellence of this team, I'd like to highlight 3 examples of how we've rapidly delivered transformational medicines to patients, starting with Lorbrena, which is our third-generation ALK inhibitor for metastatic non-small cell lung cancer. Lorbrena is a small molecule that was discovered by Pfizer scientists using state-of-the-art structure-based drug design. This is a medicine that took less than 5 years in development, significantly shorter than the industry median at the time. Moving on to Alrexfio, which is our BCMA-targeted bispecific antibody development for multiple myeloma. Alrexfio was first developed by Pfizer scientists, and we significantly accelerated the clinical development timelines. The start of the first pivotal trial, Magnetism-3, to Alrexfio's first FDA approval took less than 2.5 years. And lastly, moving to Padcev, which was developed by legacy Seagen scientists in partnership with Astellas. The recent expansion of Padcev to locally advanced or metastatic urothelial carcinoma was a tremendous feat in speed. Receipt of the top-line report from the groundbreaking EV302 trial to FDA approval took less than 3 months. And this is a remarkable feat in terms of timelines and a testament to what we believe is Padcev's groundbreaking clinical value, but also the unbelievable talent of the team that delivered Padcev to the FDA. Pfizer is consistently ranked by the Center for Medicinal Research on multiple cycle time metrics, and the latest metric from them places as number 2 in the industry for oncology. And importantly, across combined legacy Pfizer and legacy Seagen, we have secured 8 FDA approvals for 8 cancer indications in the last 3 years alone. These examples of rapid execution are great representation of what Pfizer and Seagen were able to achieve as individual companies. But together, we believe our strengths are multiplied. It is also critical to have the resources and scale to deliver these breakthroughs to patients globally. Pfizer has an extensive global manufacturing footprint with 10 of our sites manufacturing oncology products on 3 continents compared to Seagen's 1 manufacturing site. This robust internal network is critical to ensure agility and responsiveness and puts us in a solid position for growth as our portfolio expands. In fact, we have 6 times the capacity for vial volume for ADC production to what Seagen had alone. We also have 7 times more bioreactors for biologics. And we have supply channel presence in over 100 countries, enabling us to reach large patient populations around the world. Another example of our amplified impact is through our medical and commercial organizations. We have increased in size and numbers. We have tripled the size of our customer-facing colleagues in the US compared to Seagen alone. And Pfizer has commercial footprint in more than 100 countries compared to 17 for legacy Seagen. We have cross-trained now our field force, which means we should double our share of voice in key tumor areas. But just increasing size isn't everything. As both Pfizer and Seagen have been building our leadership in oncology for many years, we have each built strong relationships with physicians and customers in our key tumor areas. Our newly combined presence across both medical and commercial will expand the breadth and depth of our engagement with healthcare providers in both academic and community settings. And lastly, we have the ability to leverage Pfizer's large-scale medical and commercial platforms and capabilities, including our Chief Marketing Office, to ensure a surround sound approach with digital channels, analytics, and tools. Let's now move to our strategy. To guide us, we have a new impactful strategy in place. We are focused on 3 main modalities where we have significant depth, world-class capabilities, and expertise: small molecules, antibody-drug conjugates, and bispecific antibodies, including other immuno-oncology biologics. This gives us the ability to combine and adapt modalities to improve outcomes. For our therapeutic areas, we're building on the cancer types where we have already established presence and where we can further develop our leadership: breast cancer across hormonal subtypes, genitourinary cancer, including prostate, and urothelial cancer, including bladder cancer, hematology oncology, including multiple myeloma and certain lymphomas like Hodgkin's disease, and thoracic cancers, which include lung and head and neck cancer. Within these areas, we're initiating parallel earlier line trials where the addressable population is the greatest, aiming to both accelerate a new wave of breakthroughs and move approved medicines into earlier lines of treatment. We will continue to also develop targeted therapy opportunities where we believe we can have a true impact. For example, BRAF positive or HER2 positive colorectal cancer. You will hear much more about our tumor-specific strategies from our speakers today. Through our strategy, we believe we are positioned to drive transformational global impact through 2030 and beyond. We have the potential to increase the number of blockbuster medicines in our portfolio from 5 today to 8 or more in 2030. We shall provide insight into these 8+ opportunities today. With our R&D focus, we anticipate having a 10-fold increase in the proportion of revenue from biologics by 2030, presenting potentially more durable revenue opportunity and potentially improved outcomes for patients. These will set the stage for our ambition to double the number of patients treated with our medicines by 2030. We will share further detail of how we plan to achieve these goals through our presentations today. To close, I'd like to highlight our overall portfolio. As you can see, we have a deep and diverse pipeline spanning our core modalities and key tumor areas that underpin our strategy. You will hear more about each of these tumor areas and many of these exciting pipeline opportunities during our presentations today. And with that, we will now transition to our next session on genitourinary cancer, beginning with a reminder of what drives each of us each and every day.
Seeing your mom in pain is one of the hardest things I've ever in my entire life experienced. The pain that she experienced in her back was like mid-back all the way down to the low back, hip area, and then spread around her stomach. We were able to get my mom in the very next day for an MRI, and they had findings. They knew that she had this mass around her pancreas. She had multiple places in her spine. The doctor came back and told us that it was metastatic urothelial cancer. I will never, ever forget June 8th, one of the worst days of our family's life.
And I'm lucky I had my family right by my side through it all. I wish everybody could have an advocate like my daughter. She's the love of my life, yeah. I'm most grateful for having the family I have going through this. Then I think to myself again, how lucky am I?
Prostate and urothelial cancer are two of the most common cancers impacting patients in the United States, both with significant unmet needs. In prostate cancer, close to 300,000 new cases and 35,000 deaths are projected in the US this year alone. And with urothelial cancer or bladder cancer, up to 83,000 new cases are expected this year and 17,000 deaths. There continues to be obviously high unmet need for both types of cancers. In turn, there's a lot of activity in this space, and with many of the industry activity pursuing new treatments for both prostate and bladder cancer. The prostate cancer market is projected to increase to $26.5 billion by 2030, and the urothelial cancer market is projected to more than triple to $17 billion by 2030. Pfizer has a long heritage in improving outcomes for people living with genitourinary cancers, and we now have an industry-leading portfolio with many exciting opportunities ahead. Starting with bladder cancer, our portfolio is anchored with the groundbreaking benefit of Padcev in patients with locally advanced or metastatic tumors, with opportunities to move to earlier lines of treatment with ongoing phase 3 studies in muscle-invasive bladder cancer. In metastatic disease, we are developing a novel HER2-directed ADC, disitamab vedotin, which has shown very promising early clinical data. The ongoing pivotal program is evaluating disitamab vedotin in a subset of patients with HER2-positive disease, both high expressors and low expressors, with planned registrations in second line as monotherapy and in first line combined with pembrolizumab. Finally, in non-muscle invasive bladder cancer, we are evaluating the combination of sasanlimab, our subcutaneously administered PD-1 antibody, with BCG to improve outcomes for patients with high-risk non-muscle invasive bladder cancer. To share more about this portfolio, I'd now like to introduce two of our speakers for this session. First, I'd like to formally introduce Roger Dansey, who is our Chief Development Officer. Roger joins us from Seagen, where he was President of Research and Development. Previously, Roger was Therapeutic Area Head of Late-Stage Oncology at Merck, prior to that, Clinical Development Leadership at Gilead and Amgen. Roger holds an MD from the University of the Witwatersrand in Johannesburg, South Africa, and he brings unmatched oncology clinical drug development experience to Pfizer, as well as the industry gold standard for executional excellence. And then I'd also like to welcome Dr. Thomas Powles, who is Professor of Genitourinary Oncology at the University of London and Barts Cancer Centre. Dr. Powles is the head investigator for the Padcev EV302 trial and a world-renowned urological oncologist. We're happy to have him here today to share his perspectives on the exciting data from Padcev and bladder cancer, as well as additional medicines we have in development for bladder cancer. I'll now turn it over to Dr. Powles. Thank you.
Thanks, Chris. It's really kind of you. Thank you for inviting me in. It's nice to see you. Chris asked me to come today, and I jumped at the chance. I live in London, you can see by my accent probably. And the reason I came was because I think we are at a pivotal moment in oncology. We've not been at a point like this for some period of time. I was involved in the immuno-oncology transformation and partly involved in the targeted therapy transformation. I think we're going through one of those transformations at the moment, and I wanted to describe what my views are of those. I'm very lucky in that respect. I've led 23 big randomized trials in my life. Some have been successful, most have not. But I'm going to describe today the one that has been most successful, which was presented recently at ESMO. Padcev, enfortumab vedotin is what I call it, is an antibody-drug conjugate. It targets Nectin-4. Nectin-4 is expressed on almost all urothelial cancers. And patients with advanced urothelial cancer have a really poor outcome, median survival of about a year, historically. Padcev it binds to the surface and is internalized rapidly. It is then degraded by the lysosome. And from there, MMAE, the microtubule disrupting agent, is activated. And at that point, it triggers apoptosis. It also causes cell cycle arrest through the endoplasmic reticulum. There's then immunogenic cell death also. So that's the second mechanism. And thirdly, it's important to recognize that MMAE is permeable and can cross the barrier into other cells and trigger another apoptotic process. The drug itself, therefore, is a really attractive target for urothelial cancer, but potentially other cancers also. This is the study which I led, and I think it was, at the time we designed it, we felt it was ambitious. It turned out that that wasn't the case. We were right to be ambitious. When we designed it, we knew that enfortumab vedotin was associated with responses in patients who were heavily pretreated. We know that, well, I felt it was probably the most active single agent that we had. But you might be aware that we'd struggled to combine drugs with immune checkpoint inhibitors in urothelial cancers previously. So we did phase 2 work, and that phase 2 work showed response rates in advanced disease of 70%. That was a little bit unprecedented in urothelial cancer. We're used to about 40%. But phase 2 data can be misleading. And so it was important to do this large randomized phase 3 study. The randomized phase 3 is a classic 850-patient, first-line metastatic urothelial cancer, enfortumab vedotin, Padcev, plus pembrolizumab, Keytruda, the two drugs together, versus standard chemotherapy. During the conduct of the trial, the control arm changed. We allowed the introduction of avelumab. 31% of patients got avelumab in the control arm. That's actually quite a high proportion of patients. The endpoints, dual endpoints, progression-free survival and overall survival, with some secondary endpoints around safety and response. You can see from here, these are the primary endpoints of progression-free and overall survival. And they're overwhelmingly positive, with a 45% reduction in the risk of progression or death and a 53% reduction in the risk of death. These are by far the most impressive data we've ever seen in urothelial cancer. In fact, it's the first time we've beaten frontline chemotherapy, Gemcis or Gemcarbo, in urothelial cancer. And to beat it by 53%, the reduction of risk of death at that proportion was unprecedented. The control arm actually performed really well in this study. Historically, as I said, the median survival going back in other trials like Danube was more like 12 to 14 months. So this really was exceptional data. It's also worthwhile noting the CR rate, complete response rate, was 30%. We've never seen anything like that before. And that's why we're so excited about moving this into the perioperative setting. It's important to also recognize that in this study, we enrolled all comers. We enrolled patients irrespective of their PD-L1 status, cisplatin eligibility, which I think will become a thing of the past, but we've got very obsessed in urothelial cancer with whether a patient can have cisplatin or carboplatin. It's my feeling that the drugs have more similarities than differences. Other people disagree with me on that issue. But this was an all-comer study for the entire population. And it's likely because of these results that historically not all patients have been offered chemotherapy in urothelial cancer. The outcomes have been modest. It's quite toxic. I suspect the pie will increase. I think more people will be offered EV-Pembro than were previously being offered platinum-based chemotherapy. Adverse events are a really important part of everything that we do. They're super important for patients too. EV-Pembro was given until progression, platinum-based chemotherapy, given for 6 cycles. You can see here, actually, the grade 3 or 4 adverse events were less frequent for EV-Pembro than for platinum-based chemotherapy, 70% versus 56%. My experience of platinum-based chemotherapy and of my colleagues is not fantastic. It's associated with quite bad renal toxicity. In fact, if it was developed tomorrow, it probably wouldn't get off the ground because you've got to give IV fluid beforehand for 4 hours, then you give the platinum. We had to develop 3 antiemetic drugs to control the nausea. Neutropenic sepsis. I've got a patient at the moment in our hospital who's on their 3rd cycle, the cancer's growing, their neutrophil count 0.1, they've got infection, they're on IV antibiotics. It's a complicated regime to give. Enfortumab vedotin is different from that. It is associated with adverse events, skin toxicity in the first 3 or 4 cycles, and peripheral neuropathy, which increases at cycle 6 to 12. My experience, my personal experience of this, and I spend a lot of time speaking to people in rooms not dissimilar to this, about adverse events of drugs, is that it's manageable. In fact, I think it's easier to give than platinum-based chemotherapy. I think dose delays and interruptions are required, and early recognition of new toxicity is really important. When we go to med school and oncology school, we're taught for years about adverse events of chemotherapy, and then when new drugs arrive, we're supposed to just pick them up immediately. I think we need to think more carefully about that. But I think this is a tolerable regime. I think it's going to be very widely used throughout the world. The data made a huge splash at ESMO. I was lucky to present it. I made less of a splash, but the data was amazing and spoke for itself. It got 2 standing ovations, which was a rather unusual thing and made me quite anxious at the time. Why did it make such a big splash? It made such a big splash because we've never seen something like this before. And like pancreatic cancer, I think when we beat gemcitabine in pancreatic cancer, it'll be a similar moment. It's taken such a long time, and it's been so frustrating to see these patients come, struggle, and then die of their disease. That's just the first part of the story, because the story in urothelial cancer is going to continue. And I got a feeling the story might get better. We really need to cure more of these patients. We don't yet know about some of the durability issues of EV-Pembro. I think the median overall survival will turn out to be about 3 years at 36 months. There's a bit of instability in that curve. But the CR rate of 30% is really intriguing. I have treated maybe 20 or 30 patients with a combination. And we have patients who we've given 4 or 5 cycles to, some 8 or 9, and they've gone into durable remissions, and we've stopped some of the drugs in some of those patients, and the cancer's not come back. If we give these drugs to patients at the time of their operation, when half of them are cured with cystectomy alone, with or without adjuvant nivolumab, I think we might be able to increase the cure rate further, and I think we might be able to cure some of these patients with systemic therapy alone. And I hope it's going to be 30, 40, dare I say it, 50%. I don't know what's going to happen. But I'm very excited about these two trials, one in cisplatin eligible, one in cisplatin ineligible. One of them's enrolling, one of them's completed enrolling. You can see here the dates for 26 and 27 when they're expected, but we'll probably get some interim analysis results potentially before then. Disitamab vedotin is a drug which I'm also excited about. I've got personal experience of this drug as well. We've treated about 20 patients, and I can tell you it's also a very active antibody-drug conjugate. Disitamab vedotin has a mechanism of action which is it's targeting HER2. It's distinct from some of the other HER2-targeted therapies. It's actually internalized more quickly than trastuzumab, and the reason for that is it binds to a different epitope. It also has MMAE as a payload, so the mechanism of action in terms of the microtubule disruption, the immune component, and potentially a good partner with immune therapy, but also the bystander effect obviously applies as well. And there's a table there looking at other HER-targeted therapies and how this may compare. This drug's approved actually in China, and it's used in gastric cancer, and it's also used in urothelial cancer. As I said, it's a HER2-targeted therapy. It looks like that there is some enrichment in those patients who heavily overexpress the biomarker. But you can see actually here, the vast majority of patients overexpress HER2, and on top of that, the response rate is about 50% in heavily treated urothelial cancer. Now remember that enfortumab vedotin was 40%. So now, I'm not a great one for comparing phase 2 studies, but I'll just say that this drug's active. We've given it to our patients. It's active. The adverse event profile, because it's targeting HER2 and not Nectin-4, it's a distinct toxicity profile, perhaps less skin toxicity, in my opinion. The high rate of durable responses that we've seen with monotherapy is actually increased again when we combine it with an immune checkpoint inhibitor. This is toripalimab, not pembrolizumab. This is Chinese data again. And you can see here, both from the waterfall plot and the spider plot, that there are these deep, durable responses. I think the point that I'd kind of like to make is that I'm not convinced that all partnerships with ADCs and immune checkpoint inhibitors is going to be synergistic or additive, but it looks like MMAE may be a good partner, a good payload partner. We saw that in EV302, and we may be seeing that with disitamab vedotin also. As you can imagine, there's an ambitious plan for disitamab vedotin. Chris described his ambition, which I'm bought into. I'm also bought into the strategy for this agent. As a single agent, it has an accelerated approval with, has breakthrough status with the FDA. But it's also being explored in a phase 2 study to confirm that monotherapy activity. Our site's taking part in that study at the moment, and we're enjoying using the drugs, and we're seeing deep responses, as I said. It's also being explored in a frontline randomized phase 3, a trial design not dissimilar to the EV-Pembro trial, which I've just previously showed you. Sasalimab is a PD-1 inhibitor. It's given subcutaneously. It was developed subcutaneously, which is distinct from the other immune checkpoint inhibitors which have been developed intravenously, although some have data, or more recent data, subcutaneously. It's a potent PD-1 inhibitor. It has high receptor affinity, and it's a drug which is being explored in non-muscle invasive urothelial cancer. I described advanced disease, and I talked about enfortumab vedotin and pembrolizumab, and disitamab vedotin. I talked about that muscle-invasive setting where we do the cystectomy to cure patients, and we hope that by giving EV-Pembro neoadjuvantly, we're going to cure a significant proportion of patients. And now, earlier in the disease, non-muscle invasive urothelial cancer, where patients normally just have cystoscopy to scrape away their cancer, the standard treatment for 40, 50 years, perhaps longer, has actually been BCG therapy. It's an immune therapy. No one knows exactly how it works, which I think is intriguing. But if you go on PubMed, you'll find 100 different ways in which it may work. But you also know that in this non-muscle invasive space, pembrolizumab currently has a license, but in those patients whose cancers have progressed after BCG. This is a more ambitious plan than that. This is to go earlier in this high-risk population. BCG is a control arm. BCG is given as an initial therapy and then a maintenance therapy for a period of time. You'll be aware there's a BCG shortage from a global perspective. In this study, it's 3 arms. It's BCG plus sasalimab, BCG plus sasalimab, but no maintenance period of the BCG, and then BCG alone. I think this is an exciting study. We're hoping to see this study read out in the not-too-distant future. It's an ambitious plan, and as Chris described earlier, the Pfizer group has a very ambitious strategy. I'm now going to turn it over to Roger to do prostate cancer. Thank you very much for your attention.
Okay. Afternoon, everyone. It's great to be here, and I would like to just reiterate what Chris said from a sort of, an ex-Seagen side. I'm extremely excited. I mean, we have 2 organizations that already are at a level of high excellence. We've brought them together. This idea of best of both is extremely appealing. And more importantly than anything else is the portfolio is really, really meaningful in terms of what we can potentially do to help people. And I think that's our North Star, that's our driving force in terms of why we do this every day, is to make a difference in patients' lives. And certainly for me, it's very exciting to see all of these programs, great science with good people. Hopefully we'll bring forward some great medicines. So turning our attention to prostate cancer, which really can be broadly divided into castration-resistant and castration-sensitive disease, with actually large addressable populations across this continuum of disease. Our portfolio is anchored by Xtandi. It's the only androgen receptor signaling inhibitor which is actually approved across the prostate cancer continuum. As we seek to further improve outcomes for men with prostate cancer, we are building on that foundational drug, Xtandi, with the next generation of combinations. We have two novel combination approaches, Talzenna, which is our PARP inhibitor, and mevrometostat, our EZH2 inhibitor, both of which are being combined in development trials with Xtandi. And these two will be the focus of my presentation. However, before we get there, I'd like to remind you just what an advancement Xtandi by itself has been in prostate cancer. I think you can appreciate when you look at the weight of evidence on this slide and demonstrated by all the graphs showing improvement in progression or metastasis-free survival in all disease states. To emphasize how meaningful this therapy has been, and sort of analogous to Tom talking about the impact and the size of the treatment effect with Padcev and Keytruda, control of disease progression varies from 60 to 80%. And in addition, the gold standard outcome, which is overall survival, there's an overall survival benefit in every disease state where mature results are available. It's really quite remarkable. And actually, as recently as November of last year, Xtandi gained approval in a new population, which is represented by the results of the EMBARK trial on the far right of the slide. Xtandi now offers a new option for men with non-metastatic castration-sensitive prostate cancer with biochemical recurrence at high risk for metastasis. Moving now to Talzenna. Talzenna is building on the Xtandi backbone by targeting inhibition of PARP, a pathway that synergizes with androgen receptor inhibition. Approximately 30% of men with prostate cancer harbor alterations in homologous recombination repair genes, and these alterations are particularly sensitive to inhibition of PARP. In addition, patients whose tumors harbor a gene alteration, the HRR type, generally have an inferior prognosis compared to those who do not. And so this group of patients represents a truly high unmet need, both in castration-resistant and castration-sensitive disease. Results of the phase 3 trial of Talzenna in combination with Xtandi and the TALAPRO-2 trial have led to global approvals. And Talzenna is now indicated in the United States for men with HRR gene alterations and in the European Union for all patients with metastatic CRPC. We await the results of the final survival data mid-year, that's this coming mid-year 24, at which time we will evaluate whether these results could be used to support additional regulatory filings for this combination. Moving now to TALAPRO-3. TALAPRO-3 is evaluating the benefit of Talzenna plus Xtandi for patients with HRR gene alterations in this earlier treatment setting, metastatic CSPC. This trial has actually completed enrollment, and we expect the first data readout in the second half of 2025. If positive, this combination has the potential to meaningfully prolong progression-free survival, which should lead to extended treatment duration compared to the metastatic castration-resistant setting. Moving on now to mevrometostat, which is our EZH2 inhibitor. EZH2 plays an important role in controlling gene activity and is often associated with prostate cancer cell proliferation. Mevrometostat in combination with Xtandi has demonstrated promising synergy in preclinical models, as you can see on the slide. And this promising preclinical data supported the initiation of a trial evaluating mevrometostat and Xtandi in heavily pretreated patients with metastatic CRPC. And we believe that this molecule is potentially the first EZH2 inhibitor has the potential to be approved in prostate cancer, obviously subject to the trials being positive. The updated results of the dose escalation portion of the phase 1 study are shown here. And the combination of mevrometostat plus Xtandi has delivered a meaningful prolongation of radiographic progression-free survival in patients who have already failed Xtandi or abiraterone treatments. For the post-Xtandi patients, a radiographic PFS of 11.7 months was achieved. You can see that on the right side of the table. And this benefit is even more pronounced in patients who have only received prior abiraterone, with a median radiographic PFS of 17.1 months. Both of these results are extremely encouraging, as the median RPFS for patients who previously received either Xtandi or abiraterone and treated with available therapies is only 5 months. Of note, the most common treatment-related AEs in this study were diarrhea, dysgeusia, and anemia. Based on the strong signal, we are planning to initiate 2 pivotal studies in metastatic CRPC later this year. The first, phase 3, will evaluate mevrometostat plus Xtandi compared to either Xtandi or docetaxel in patients who previously received abiraterone. In parallel, and that's on the right side of the slide, we are planning to start a second phase 3 study, which will compare mevrometostat plus Xtandi versus a placebo base with Xtandi. And this is in patients who are treatment naive with metastatic CRPC. So bringing the entire genitourinary cancer program together, and as you've heard from the presentation from Chris, from Tom and from myself, we are really excited by the opportunity for Pfizer Oncology in GU cancer. In the near term, we have these 2 phase 3 trial starts with mevrometostat, which I've just walked through with you. We also expect near-term data readouts for Talzenna and Xtandi with that final OS data later in the year. The trial that Tom ran through on sasanlimab with non-muscle invasive bladder cancer combined with BCG, a really exciting trial. That's a proximal readout. And disitamab vedotin, our other approach to bring a vedotin-based ADC to bladder cancer in second-line HER2-positive and HER2-low urothelial cancer, metastatic urothelial cancer. So we have a lot of near-term data readouts, which I think are pretty exciting, potentially very exciting. Longer term, obviously, we'll have numerous readouts from registration trials. And again, I think as Tom emphasized, the PADCEV and KEYTRUDA story is not stopped. There's the top-line muscle-invasive bladder cancer data, which will be available potentially as early as next year. And this has tremendous potential, just based on PADCEV and KEYTRUDA's track record, both of these highly active agents. There's a strong possibility these trials will be positive, and with the potential for this combination to really become the new standard of care in a large patient population. In addition, we anticipate the readout of disitamab vedotin plus KEYTRUDA in that frontline metastatic HER2-positive urothelial cancer, as well as the pivotal trials of mevrometostat plus Xtandi in metastatic CRPC. So with our innovative portfolio across prostate and urothelial cancers, GU has the potential to emerge as the largest growth driver among our tumor areas of focus. And I believe we're well positioned to reach a significant portion of the over 150,000 addressable patients in the US in the next decade. Thank you, and I'll turn this back to Chris to continue with our tumor area presentations.
I started, I was just in for my yearly physical. I said, well, I had this kind of unproductive cough for a while that I can't seem to shake. Got X-ray that showed something. Then he decided to send me to a pulmonologist, and she did some more testing. Lung cancer. Said it was stage 4, already metastasized. I got biomarker testing after they got the tissue. It took 3 different surgeries before they got enough to do the testing, but it also came back as an ALK positive gene.
Having hope that there's something there that can help, it's the biggest thing that you can have as a cancer patient, as a cancer family. Every moment matters.
Thank you. In thoracic cancers, we're advancing portfolio across both lung cancer and head and neck cancer, as well as lung as the #1 cause of cancer deaths globally. There were approximately 280,000 new cases of lung cancer in 2023, with approximately 130,000 deaths. Head and neck cancer occur in the larynx, throat, lips, mouth, nose, and salivary glands. In 2023, approximately 67,000 new cases of head and neck cancer were diagnosed in the US, and there were approximately 15,000 deaths. There's a high, very high unmet need for new options for curative intent or to prolong survival. The lung market is very active today and projected to increase to $45 billion by 2030, and the head and neck cancer market to $3 billion by 2030. I'm now making to introduce Dr. Megan O'Meara, who is our head of early clinical development. Megan joins us from Seagen, which she joined in 2011 and most recently was head of clinical development at Seagen. She's a board and certified medical oncologist. Megan received her MD from the University of Arizona and then internal medicine residency at the University of Washington. She completed her medical oncology fellowship at the Fred Hutchinson Cancer Institute in Seattle and also completed the postdoctoral fellowship at the University of Washington. Thank you, Megan.
Thanks, Chris. Good afternoon, really everybody for joining today. I can tell you I'm thrilled to be here and really excited to tell you more about how we're developing potentially best-in-class medicines for thoracic cancers, including lung cancer and head and neck cancer. So as Chris touched on earlier, and you can see here, our combined pipeline provides very broad opportunities for development in the thoracic space, including but not limited to the 11 medicines shown here. This robust portfolio spans Pfizer Oncology's core scientific modalities, with clinical stage assets across small molecules, ADCs, and immuno-oncology biologics, which also includes bispecifics. On the left, you can see our small molecule programs. This includes our SHP2 and see below and our oral STING agonist, which are both in phase 1. And then you can see below our approved medicines, including our third-generation ALK inhibitor Lorbrena and combination therapy Braftovi-Mektovi, which was recently approved for adults with metastatic BRAF mutant non-small cell lung cancer. In the middle are our innovative ADC programs. This includes B6A, which is an integrin beta-6 directed ADC that was recently assigned the non-proprietary name sigvotatug vedotin. And it recently began a phase 3 study in second to third line non-small cell lung cancer. Then below that, we also have some other phase 1 ADCs, one directed at PD-L1 with a vedotin payload, also a CEACAM5 with a topo payload. And then finally on the right, you can see our immuno-oncology biologics. This includes a novel EGFR targeted bispecific that's just entered the clinic. So today what I'm going to focus on is the progress we've made with Lorbrena, as well as our vedotin ADCs directed at integrin beta-6 and PD-L1. So first, to set the stage, I'd like to share a little bit about the epidemiology of non-small cell lung cancer. As you can see on the left, there are groups of patients with actionable genomic alterations, such as the ALK gene. Each patient's cancer is unique, and so testing for specific genomic alterations may allow for more personalized treatment. Treatments can also differ based on histologic subtypes. As shown on the right, nearly 3/4 of the disease is non-squamous and about 1/4 squamous. As a pioneering leader in precision medicine, we've developed several targeted lung cancer therapies, including Xalkori, which was the first precision medicine in lung cancer targeting the ALK gene, and more recently with Lorbrena, our third-generation ALK inhibitor, which is emerging as the potential standard of care for ALK-positive non-small cell lung cancer. I'll talk more about this shortly. And then looking ahead, you can think about our innovative pipeline of potential first-in-class therapies, which looks to maximize our opportunity in lung cancer across broader populations using a variety of innovative modalities, including novel ADCs and new targets. So I'll look forward to sharing an example of this related to an ADC directed at integrin beta-6, which is a target expressed on a large majority of lung cancer. Shortly following the development of Xalkori, our expert Pfizer medicinal chemists embarked on further addressing unmet need in ALK-positive non-small cell lung cancer with Lorbrena. With a unique macrocyclic structure, unlike any other ALK TKI, it was designed to intentionally overcome ALK resistance mechanisms and also better address brain penetrance. Of note, brain metastases are particularly common in ALK-positive non-small cell lung cancer, and so I'll touch more on the importance of this shortly. As you can see on the table on the right, the green shading demonstrates the broad mutational coverage of Lorbrena against some of the most common tumor mutations that drive resistance to other ALK inhibitors. This shows Lorbraina's unique potential to differentiate in this space. And then I also want to point out, as Chris talked about earlier, that we were able to rapidly execute the development program for this medicine, leading to accelerated approval in less than 5 years after IND submission. So through the phase 3 CROWN trial investigating lorlatinib versus crizotinib, we were able to validate the rational chemistry design in the clinic. The FDA approval of Lorbrena is in the frontline setting for ALK-positive non-small cell lung cancer is based on these compelling results from the trial. As you can see here, Lorbrena demonstrated a significant class-leading 72% reduction in the risk of progression or death compared to our original molecule, crizotinib. Not only is this improvement in the outcome compared to crizotinib quite remarkable, but as you can see, the tail of the curve has plateaued, suggesting that patients could remain on treatment for a long time before they go on to progress. So as I mentioned earlier, Lorbrena was designed to better address brain penetrance. Importantly, upon initial diagnosis, up to 40% of ALK-positive non-small cell lung cancer patients will be impacted by brain metastases. As you can see on the left, Lorbrena demonstrated meaningful improvement in the reduction of brain metastatic progression in the CROWN study, with a hazard ratio of 0.08. This is rarely seen. And as you notice on the right, reduction in risk of brain mets has impact across patients with or without brain mets at baseline. In fact, if you look at the far right, at the time of this analysis, only one subject of the 112 without brain mets at baseline went on to develop brain mets on the Lorbrena arm of the study, suggesting a protective effect. Now, as shown here, there have been several phase 3 studies in ALK-positive non-small cell lung cancer that carry crizotinib control arms. Head-to-head comparisons cannot be made, but I'd like to point out that the crizotinib control arm in all 3 of the trials shown here performed similarly. Now, if you look at the experimental arms, lorlatinib appears to have performed best. As you can see on the left, the 3-year follow-up data from the CROWN study show that Lorbrena demonstrated a 73% reduction in risk of progression or death compared to crizotinib, corresponding to a hazard ratio of 0.27. Importantly, the curve is still plateaued, and we've not yet reached median PFS or overall survival. Median duration on treatment for Lorbrena was 33.3 months at the time of this analysis, with 61% of patients still receiving Lorbrena versus 8% on the crizotinib control arm. So in summary, these remarkable results support Lorbrena as the potential standard of care in ALK-positive non-small cell lung cancer. So now what we're doing is gathering the 5-year CROWN data, and we look forward to presenting what could be potentially unprecedented results at an upcoming medical conference. Okay, so switching gears, now I want to touch on our growing portfolio of innovative ADCs that have the potential to improve the lives of many patients living with thoracic cancers, including lung cancer. Sigvotatug vedotin is an integrin beta-6 directed ADC, also known as B6A. Integrin beta-6 is highly expressed across a variety of solid tumors and is typically associated with a poor prognosis. In non-small cell lung cancer, integrin beta-6 is actually expressed on greater than 90% of tumors. So you can see here on the right, immunohistochemistry staining examples of integrin beta-6 across histological subtypes. In fact, other groups have tried to target integrins for cancer therapies, but they've been limited by challenges with normal tissue target expression. Our team of researchers intentionally designed this integrin beta-6 antibody for high target selectivity, limiting binding to other integrins that are more likely to be expressed in normal tissues, potentially reducing off-target toxicity. And then in addition, just thinking about what Dr. Powell's referenced this morning with the mechanism, by pairing this target with the vedotin payload, the MMAE release leads to not only direct cytotoxicity and bystander effect, but also immunogenic cell death, leading to potentially complementary activity with immune checkpoint inhibitors like anti-PD-1s. So today, I'm pleased to show you some encouraging new data from our phase 1 study of sigvotatug vedotin. This is a heavily pretreated phase 1 population of non-small cell lung cancer patients with limited standard of care options and high unmet need. For example, just to put it in context, with docetaxel therapy, one would expect a response rate in the low teens. These data are in a subset of non-squamous non-small cell lung cancer patients across active dose regimens of 1.25 milligrams per kilogram or higher. If you look on the left, you can see the waterfall plot. If you look at this inflection point, nearly 2/3 of patients are showing tumor regression. In this phase 1 study, the overall response rate with the selected phase 3 dose of 1.8 milligrams per kilogram biweekly, which was supported by regulatory authorities, was over 30%, 31.3% to be exact. And then if you look here on the right, you can see the spider plot within this cohort. And you can see many patients had deep and durable responses with some on treatment more than 18 months. And so we're continuing to follow the data, and we're looking forward to sharing more updated data at an upcoming medical conference later this year, including survival follow-up. And then now here, this is our design for our sigvotatug vedotin pivotal trial, which is now up and running. We designed this trial specifically based on the learnings from our phase 1 data. And you saw per the previous slide that in this data set, we saw the best clinical activity in the non-squamous subset. Now, as I noted in my upfront slides, nearly 3/4 of patients with non-small cell lung cancer have non-squamous disease. So given the favorable activities observed in the subset from our phase 1, we'll be focusing on second to third line, taxane-naive, non-squamous, non-small cell lung cancer. Patients will be randomized to sigvotatug vedotin or docetaxel, and the primary endpoints of the study are objective response rate and overall survival. And I'm pleased to say the study is now recruiting, it's up and running, and we anticipate a readout in 2026 or 2027. So let's move on to head and neck cancer now, where there's also a substantial unmet medical need. We see significant opportunity to improve upon the standard of care, particularly in patients with unresectable disease in the locally advanced or metastatic setting, which you can see here in the pie chart for the 3 lighter blue colors accounts for approximately 60% of patients with head and neck cancer. We have many innovative programs that we believe can make a difference for people living with head and neck cancer, and I'm going to focus on one of them today, our PD-L1 targeting ADC. So shown here is our first-in-class PD-L1 targeting vedotin ADC. It's interesting, prior to initiating this program, we considered the target to be interesting for multiple diseases, just given the high expression of PD-L1 on tumor cells. And given this, we questioned why haven't others come up with PD-L1-directed ADCs? And then in the lab, when we conjugated our vedotin drug linker to commercially available PD-L1 antibodies, they actually didn't work well as ADCs. They don't really internalize. And so our talented team intentionally created a PD-L1-directed ADC with excellent internalization. So as shown on the right, when we compared the extent of internalization to other PD-L1 backbones, including atezolizumab, avelumab, or durvalumab, our antibody achieves much better and much faster internalization. And in addition, we've been able to demonstrate preclinical activity across the PD-L1 spectrum, even in PD-L1 low-expressing tumors. So looking ahead, again, we see opportunities to leverage this vedotin immunogenic cell death mechanism and also explore potential combinations with immune checkpoint inhibitors with this ADC. So here, I'm excited to show you what is the first disclosure of clinical data from our phase 1 study, which is demonstrating interesting initial efficacy. So if you look on the left, you see the data from the head and neck subjects in dose escalation greater than or equal to 1.25 milligrams per kilogram. At the inflection point of the waterfall plot, approximately 2/3 of patients are showing some tumor regression, with a confirmed objective response rate of 21% and an unconfirmed objective response rate of 50%. We continue to follow this data closely. And to put it in context again, these are very heavily pretreated patients with limited options. In this particular data set, the patients here received a median of 4 prior lines of therapy, and all of the patients' tumors have previously progressed on immune checkpoint blockade. So in this setting, to put it in context, one would expect single-digit response rates with available therapies. And then you can look on the right and you can see the spider plot, which shows ongoing responses, including patients currently approaching 8 to 9 months on treatment. So this is early data. We plan to present additional data at an upcoming medical conference later this year. We also plan to initiate PD-1 combo studies in the frontline setting as part of the phase 1 trial. So I've been excited today to share some data from 2 promising ADCs in our thoracic pipeline, one targeting integrin beta-6 and the other targeting PD-L1. We've generated consistent preclinical data sets supporting that vedotin drives immunogenic cell death, and we see potential clinical synergy with anti-PD-1. So as you can see here, enhanced clinical benefit has been seen across multiple combination studies exploring vedotin ADCs with anti-PD-1s. And that includes with the recent EV302 pivotal data in urothelial cancer with Padcev and Keytruda that was just presented by Dr. Powell's. So we're making nice progress advancing these efforts with PD-1 combos ongoing for sigvotatug vedotin and planned for the PD-L1 ADC in the phase 1. And so this will help us drive development into earlier lines of therapy. So given the unmet need across thoracic malignancies worldwide, Pfizer is committed to developing the next generation of potential breakthroughs in this space. Today, I was able to show you a significantly expanded thoracic portfolio with encouraging data for Lorbrena, sigvotatug vedotin, and our PD-L1 ADC. Now, looking ahead, we're excited about the potential catalysts shown here. In the near term, this includes that we've actually just initiated enrollment of our phase 3 study exploring sigvotatug vedotin in second to third line non-small cell lung cancer. Soon we'll be sharing our 5-year data from the Lorbrena CROWN study. And then we have the evaluation of data-driven opportunities for our PD-L1 ADC in PD-L1 expressing tumor types, including head and neck cancer. And then looking out even longer term, we're looking forward to broader opportunities in the thoracic space with initiation of a phase 3 study exploring sigvotatug vedotin in frontline non-small cell lung cancer. And then looking a little further out, we're very excited about data readouts from the phase 3 study ongoing of sigvotatug vedotin in second to third line non-small cell lung cancer and the planned first line non-small cell lung cancer study. So putting this all together, Pfizer is well positioned to bring this innovative pipeline of potential breakthroughs to a significant proportion of the over 150,000 total addressable head and neck and lung cancer patients in the US across the disease continuum in the next decade. Thank you. Now I'll turn it over to Chris for Q&A.